Hello, good morning, everyone. Good to see you all again. How are you? Good. I hope you enjoyed the evening yesterday. I hope you enjoyed the workshop yesterday, by the way. Welcome to the second day of our workshop. We will continue with an overview on activities which have been performed in the 30 years, during the 30 years of joint US-Russian collaboration in the Southern Urals. And afterwards, we will discuss or present and explain why we as ICRP feel the results we heard are important. Before doing so, I would like to introduce to you Win Tusar. She will moderate the session this morning. Win is from Nagasaki University. She is a third-year PhD student, and her research focuses on the human aspects of radiological protection in the recovery phase after a nuclear accident. And as it happens today, it is her first day as an intern in the scientific secretariat of ICRP. So this is her first duty. So please welcome Win. And Win, please, the floor is yours.
And thank you so much. Good morning. If I make any mistake, excuse me. And start with the first speaker. I would like to invite Smith Miller to present additional complemented projects and other JCCRER supported science. And welcome Scott Miller, sorry.
Good morning, everybody. A little preface to these talks I'm going to be giving this morning. When we were going through the organization of what we thought we would present, we thought it was kind of important to present at least a brief scientific overview of all the projects that received a JCCRER number, even though they're not all they haven't talked about them here today. I was not directly involved with any of these, so I apologize in advance to the PIs. But I was involved with Project 2.4 that did interface with all of these projects. So we were aware they were going on. We interacted with the investigators periodically. And what I'm going to present today is just the original scientific objectives that were proposed when these were presented. And then I took the liberty to go through and pick out some highlights from the published literature that they presented. On most of the slides, I will have references, and if anyone wants those references, I have PDFs. Get my email and I'll send those to you. Although a lot of them are easy to find by typing in any search engine. The two primary publishing authors, the Russian author and then the other author, and it'll pop up on Google or PubMed or whatever. Let's begin with Project 2.1. I'm going to go in order, in sequence, even though that was not necessarily chronological, but let's begin with Project 2.1. I might add that all of these projects were what I would term foundational projects. They were either providing an infrastructure for us or sorting out methods and techniques or databases, or even doing some experiments in molecular biology to look at the feasibility and practicality of using these tissues, these resources. Next slide. I guess I control these, don't I? The first program was, the PIs were Valentin Hokurikov from, at that time it was called First Institute of Biophysics, FIB1. Later became SUBI, Southern Urals Institute, and Ron Philippi from the US Transuranium Registries. The stated objectives of this program were to conduct joint analyses between the US TUR and what was then called the DRIMA, the Dosimetry Registry at Mayak, but that really represented Mayak and SUBI together, that combined all the various databases that existed in those organizations. They wanted to see the comparability. They wanted to compare techniques. They wanted to do upgrading and things like that. And the intention was to, of course, develop and improve dosimetry models. Here's a picture of Keith Eckerman and Valentin Hokrikov in front of an old alpha counter that was homemade. By the way, Valentin Hokrikov, like Nina Kochernikova, was a force of nature when I first met him back in the mid-1990s. Actually, I met him on another project associated with NATO, but he probably knew more about the biology of plutonium in humans than anyone on the planet. And between he and Nina and a few others, it was remarkable what the resources and they had, and he was a fabulous collaborator over the years. The accomplishments, they basically wanted to compare the techniques with the idea of establishing common procedures, looking at reagents and various things. The first surprise that the USTUR investigators had is when they assembled this cohort of Mayak workers and they assembled a group of USTUR participants, they immediately found that the liver concentrations in the Mayak workers were on average over 200 times the plutonium that they found in the higher dose rate people or dose plutonium concentrations in the USTUR. So already they recognized real soon that they were dealing with some really significant plutonium concentrations. They established uniform radiochemical methods and equipment. There was upgrades in equipment over the years. They exchanged samples to look at solubility or to compare techniques. And then they also looked at Valentin's Hokrikov's. He had an in vitro solubility assay that he had used to look at the solubility of the various industrial compounds of Mayak. And then he extrapolated that to transportability in the lung. There was some early work done by Clara Suslova on liver diseases related to plutonium that they looked at. And then they developed in the so-called FIB1 model, which was the early model, lung clearance models. This project was later merged with Project 2.4. Again, references, if you want them, I can get them to you. Project 2.3. This was a study Bruce mentioned yesterday. This was funded at least partially by the Nuclear Regulatory Commission. Nadia Kladnikova was a Russian Federation investigator, and Greg Klaykamp, and there was other investigators involved, but these were the two primary individuals. The stated objectives were to look at and validate, look at the acute and chronic effects of radiation, everything other than cancer. And this is a topic that came up yesterday, the deterministic, non-cancer deterministic effects. The NRC was interested in using this to test their regulatory models. Some of the main accomplishments from this study is they developed a cohort of about 600 workers initially who had both external and internal dosimetry, and they had the work history and medical records. And this project was particularly interested in looking at clinical chemistry or blood chemistry markers and other clinical indicators to see if they could better define and better diagnose these deterministic effects. And they did that looking at a lot of blood cell counts, and they found that blood cell differentials counts improve the diagnostic classification criteria. We have plutonium pneumosclerosis on there. That was one of the early deterministic effects that was noted in the 1950s, and it was published in the Russian literature for many years. And it really didn't emerge into the English literature until this project started. And we saw some slides on that yesterday with the pathology of plutonium pneumosclerosis. I have highlighted down here a reference in purple, the Azizova 2019 reference, because the deterministic cohort is now expanded to about over 2,000 workers. And that was published in 2019. It's workers with the chronic radiation syndromes. So if anyone is interested in deterministic non-cancer effects, that would be a great cohort to look at. It's been recently updated. Project 2.5 was one of these mechanistic studies that was kind of, it was when they proposed it was, can we do this? Will this work? Can we use these tissues that are heavily contaminated with plutonium to do something simple like conventional autoradiography to look at distributions of plutonium in the lung? This was done by Sergei Romanov, the director of SUBI, and Ray Gilmet and his colleagues at the Lovelace Respiratory Research Institute, which was previously ITRI, the Inhalation Toxicology Research Institute, over many years. This project was later merged in with Project 2.4. The original objectives were to see if they could use the archive tissues to look at plutonium distribution, and they use stereological and semi-quantitative techniques to look at these distributions and correlate it with histology in the images. They looked at they reported on different lung regions and areas, and the question they asked originally was, how might non-uniform distributions influence dose-related lung cancer risks? They found that plutonium in the tissues was not uniformly distributed, which we kind of knew from previous animal studies and other human studies. Over 40% of the was found in parenchyma and clearly associated with areas of fibrosis. Below is an autoradiograph that I took from one of their papers. They'd also presented this in meetings, but it showed a plutonium star in the alveolar region of a lung. They did this again using conventional autoradiography. They also found a lot of plutonium in the lymphatic tissues, in the lymph nodes. On a separate project that I did with Sergei Romanov, we did some neutron induction studies, and the plutonium concentrations in the lymph nodes, some of the pulmonary lymph nodes, was so high it literally blew my detector out. It saturated the detector. So very high concentrations were found in these fibrotic areas and in lung and in lymph node areas. We discussed the issue of immune system things came up yesterday a little bit. And this is one of the areas I personally think should be looked at further with relative to lung, is the lymphatic tissues and the lymphatic systems. And this is an emerging area of interest in biology in general. So this was an interesting project. It really demonstrated very graphically these distributions and exposures, and was really a delightful project. They also developed some new Bayesian approaches with Guthrie Miller, who was at Los Alamos at the time. Another interesting project, molecular markers of lung cancer. Again, this was taken kind of like, let's just see if these will work, if these tissues have enough damage, genetic damage or changes in molecular markers. And this was done by Vitaly Telnov at SUBI and Steve Belinsky at the Lovelace group. The initial stated objectives were, can we identify molecular markers in these tissues? The identification of molecular markers has been a long-standing quest in radiation biology, looking for shortcuts to distinguish radiation damage from other types of damage, of course. And they wanted to look at markers of lung cancer, look at some molecular mechanisms, and they used basically hypermethylation, which would suppress tumor suppressor genes, genes that were associated with cell cycle regulation and things like that. They did a kind of a cross-sectional study where they took plutonium workers, workers at Mayak who were not exposed to plutonium, like reactor workers, and then an unexposed population of non-workers. And so it was a cross-sectional type study they did. What they found, and you can see down here in the slide I took out of one of their papers, is they found a dose-response relationship to the frequency of methylation of these suppressor genes. And it's some of the details here. They did most of their work in adenocarcinomas and some work in the squamous cell carcinomas. They found that particular slide is males, and they found a similar but not statistically significant relationships in female workers, primarily because they didn't have enough to achieve statistical significance, but the trend was there. So this, they concluded that the inactivation of genes by methylation is important in cell transformation, and they thought that this could be an important pathway for plutonium adenocarcinomas. And of course, work's been done since then on all of these projects. So this was the first using the Mayak workers, which is why I think it was really one of these foundational important studies. The next interesting study, again, trying to identify radiation biomarkers, and this was done by Tamara Azizova with David Brenner at Columbia University. And here they used, the stated objectives were to develop a dosimetry-based system, biomarker-based dosimetry system that would distinguish plutonium and gamma exposures. They were most interested in being able to look at stable markers that would identify past exposures. They were doing intra and intrachromosomal aberrations, and they were using fluorescence in situ hybridization techniques. Particularly, you see in the image I took from their paper, the multifluorescence, M-band and M-fish technologies. And it very eloquently demonstrates the aberrations shown in the chromosomes. What did they find? They found that, in fact, those techniques were suitable, were valuable, and could identify intrachromosomal rearrangements in plutonium workers. They found significant excess in certain chromosomes, 1, 2, and 5 in plutonium workers compared to those exposed to high gamma rays. So they were able to distinguish initially some of the gamma exposures from the plutonium exposures. The conclusion there I took directly out of one of their papers was that stable complex chromosome translocations represent a sensitive, specific, and long-lived quantitative low background marker of densely ionizing radiation exposure in this population. They were interested in, of course, these stable markers. Again, since this time, other work has been done on this that confirms this and extends it to even different chromosomes using different technologies. Project 2.9. This was initially a short-term project, but I want to talk just briefly about this because this was really a project that started, it literally started the day that the JCCR started, because it entailed a whole lot of work initially identifying these databases, identifying the records, all of which at that time were paper in various forms in different locations. There was a large effort to microfilm these records, over 1 million records. And then the microfilm was later digitized, turned into a digital format. And then Mikhail Sokolnikov, Mikhail Goryalov from Mayak, and Del Preston, who at the time was more affiliated with RERF, Dale was instrumental in enlisting some of their experts to help construct and organize the initial databases that we had at Mayak. So in reality, it was a huge project of which very few papers have ever been published about that effort. The real challenge initially was to develop a master database, common identifiers, to be able to link all these various databases that existed. not really databases, oftentimes just paper records and masses of paper records like you can see here. We had to document these. There was an effort early on about declassification because many of these reports at the time were classified and they needed to be reviewed by the Russian authorities to release them. And this has changed over time as the databases have updated, the equipment is updated, and different platforms have been used. That kind of summarizes the accomplishments. The database people, and there's a large number, converted them to digital initially and saved them, developed common links, common identifiers, all while we were also concerned about human use ethics committees as they evolved in the various organizations that were involved, and the ability to migrate these databases to different platforms suitable for statistical analyses. The Melamid publication at the bottom describes the microfilming effort. And that is it. Many thanks to the many people involved in this. It's been an incredible experience. The picture on the left is a group of us who perhaps foolishly were standing on the lid of the Kishtim disaster, which was, that's the tank lid that blew off. And of course, as noted yesterday, it really wasn't in Kishtim, it was out on the Mayak site. And of course, the beautiful Seversk. Again, if you want references, please drop me an email. Thank you.
And thank you so much. Now I would like to invite another speaker, Dariya Oslina. She will give remotely about their biorepository, SUBI, Radiobiological Human Issue Repository, History and Perspective.
Good morning, everyone. Can you hear me?
Yes, everything is okay.
Okay. I'm very excited to present you results of our work in the Russian Radiobiology Human Tissue Repository. So let's go. Repository in Seversk is the first radiobiological repository in Russia. The collection of samples to study the health effects of ionizing radiation began at SUBI back in 1960s. And an invaluable contribution to the creation of repository was made by Soviet outstanding radiobiologist Vladimir Lemberg, Grigory Baisagolov, and Angelina Guskova. The repository collection started from autopsy tissue stored in formalin solution, paraffin blocks with formalin-fixed tissues, and slides. Since 2001, financial support for repository has been provided by U.S. Department of Energy under Project 2.8. And at that time, under the leadership of Clara Maksinova, repository began collecting samples of whole blood and its components, as well as extracted DNA. The repository collection has also expanded to include surgical and biopsy material, and today repository is a structural subdivision of the SUBI clinical department. Nowadays, the repository is collecting and storing biological material to study the effects of ionizing radiation on the organs, tissue, cellular, and molecular levels. Our task is to obtain, process, and store the biological samples, monitoring of storage conditions, obtaining and clarifying demographic, professional, dosimetric, and medical information about registrants, providing researchers with high-quality annotated biological material, and informing the scientific community about the results of repository work. Biological samples in repository are stored under different conditions depending on the type of the material. In fixing solution are stored autopsy organs and tissues and surgical or biopsy tissues. In frozen specimens are whole blood and blood components, DNA, induced sputum, saliva, and buccal cells, frozen autopsy and biopsy tissues. And in the form of paraffin blocks and slide, we store autopsy and biopsy tissues, blood and blood marrow smears, and cytogenetics preparations. For all of these operations, we have SOPs. Sample collections follows International Society for Biological and Environmental Repositories guidelines, and all samples are marked with individual barcodes. We obtain and store specimens and associated data not only from exposed individuals, but also from unexposed blood workers and from sex and age matched population living near the radiation hazardous plant. Now, repository stores 406,790 samples from the 15,723 registrants. Of these, more than 80% are Mayak workers and others are the controls. The ratio of men and women in the cohort of Mayak workers is approximately 2:1. And in the control group, as can be seen from the diagram, women predominates. Occupational diseases were registered in 339 registrants. Among them were acute radiation diseases, chronic radiation disease, occupational pneumosclerosis, combustions, dermatitis, wound, and cataract. We store samples of blood and its components from 12,000 registrants, autopsy from 1,270 registrants, and biopsy samples from more than 1,400 registrants. A significant part of the samples obtained during autopsy are paraffin blocks, sites, and I missed something. And the collection also includes samples and decalcified bones. Yes, this is. And decalcified bones presented in alcohol fixing solution. During autopsies, our staff collect samples of internal organs, brain, endocrine glands, reproductive organs, and lymphoid tissue. The quantitative distribution of registrants, depending on the type of organs and tissue, is presented in diagram. The repository database contains information on the causes of death of all registrants, as well as on the morbidity of for the entire period. The quantitative distribution of repository registrants, depending on the location of malignant tumors, can be seen on the figure. Surgical and autopsy tissue are samples of tumor and non-tumor tissues obtained during surgery or biopsy and immediately placed in liquid nitrogen. As it can be seen, women predominate among biopsy tissue donors. Soft tissue samples are stored in freezers at temperature from -75 to -80 Celsius degrees, as well as in the form of paraffin blocks and slides. As it can be seen in the tables, among malignant tumors, tumors of mammary gland and colon predominate. Among benign neoplasms of reproductive organs are most common. Blood samples were collected from 12,000 registrants. This is now 210,663 aliquots. Men predominate among registrants. The quantitative distribution of registrants, depending on the type of plant, can be seen in the diagram. The repository stores samples of whole and snap frozen blood, plasma, serum, erythrocyte, leukocyte, and lymphocytes. Also, DNA samples were isolated from peripheral blood. Today, the repository family bank stores biological samples of 1,715 registrants, forming 555 family trees. The repository database includes all data on parents' preconception doses from the beginning of their career at Mayak to time of conception, as well as medical dosimetry data. Over the years of its existence, the repository has repeatedly transferred biological material to scientific institutions for their studies in the field of radiobiology. It's a unique resource for research in radiation, cytogenetics, immunogenetics, radiation immunology, biological dosimetry, studying the mechanism of radiation-induced cancer and non-tumor effects, metabolism and distribution of radionuclides in organs of deposition. In the end, I like to express my deepest gratitude for all DOE colleagues and collaborators, especially for Christopher Loffredo, Adila Laurier, and David Gerlitz. Thanks you very much for your attention.
Thank you so much. So we invite next speaker, Chris Loffredo, who is giving the presentation about evaluations that have been done with HTR data at Georgetown. Thank you.
Can you hear me?
Yes,
yeah,
floor is yours.
All right, first slide, please. Thank you very much. Also, my thanks to the organizers of this meeting for inviting me to speak. And my colleague Dariya has just given you a very good overview of what is in there, and I won't overlap with her too much. I've been the US PI at Georgetown University for Project 2.8 since 2008. Initially, I worked with Klara Muksinova, then for 15 years with Evgenia Kirillova, and more recently with Tamara Azizova, who is the current PI of Project 2.8. Next slide, please. As Dariya just told you, the biorepository was founded quite a long time ago, and you understand the mission is to not just put tissues away, but as you see in the second bullet, really to support basic and molecular epidemiological studies on radiation mechanism and exposures. Next. So I'm going to just very briefly highlight a few of the methods that Dariya already told you. Next. The biobanking methods, two main groups, as you heard, the Mayak production workers and residents of the city that we call controls have been enrolled with signed informed consent of living donors and by the next of kin for those who are deceased. And every specimen is annotated with important information about occupational materials, demographic characteristics, dosimetry, medical information. And it went by quickly, but Dariya did tell you that the biorepository really does conform with international best practices guidelines. And in fact, it was certified by the International Society for Biological and Environmental Repositories as being in compliance with current best practices. And we're very proud of that achievement that we helped our colleagues at SUBI to achieve. Next. Autopsy tissue bank you heard about. Next one. And you also heard about the surgical tissue bank. These two are very important in supporting studies of both cancer and non-cancer outcomes. And I'll be telling you more about some recent cancer research in a few slides. Next. You heard about the blood components bank. And although it went a little bit fast when Dariya was telling you about it, this is a massive effort. You saw the thousands upon thousands of specimens that are stored. You know, each time a person donates whole blood, there's a lot of effort to separate it into all the different components that you see listed here in the second bullet, leukocytes, erythrocytes, etc. There's a lot of effort to store and track these materials. And what I wanted to mention to you also is that everything is barcoded. When I first got to SUBI in 2008, things were labeled by hand. The staff was very cognizant of the need to upgrade the repository methods, but in particular, we spent years and years supporting the barcoding of the entire collection. More than 350,000 individual items were eventually barcoded, and that's now the standard practice for all incoming specimens. Next, please. Let's go past that one. So I wanted to tell you a little bit more about the quality control and quality assurance testing. Next, please. Quality assurance is not trivial. And, you know, it may seem like you can collect tissues and just put them away in a freezer and then forget about them. But in fact, the staff maintains this material under excellent conditions. What do they do? As you see in the first bullet here, the storage solution in those containers containing formalin, they're checked on an annual basis and are replenished so that the pH is optimal for the long-term use of those tissues. We have assessed the viability of using the blocks, you know, the tissue blocks. We have looked at blocks that were stored up to 50 years in the biorepository, and I'm going to show you some slides in a minute demonstrating the quality of those materials. They're absolutely pristine. We brought a set of liver tumors and breast tumors to Georgetown University, where an expert pathologist reviewed them and confirmed the details of the histological diagnosis. And in a study that I'll talk about in a minute, we obtained DNA out of these stored tissue blocks. And 80% of those tissues had sufficient genetic material and sufficient quality of material for actually supporting molecular genetic studies. And I'm going to tell you about DNA sequencing that we've done. So these are really pristine. Next. The histological quality is so good that, in fact, the pathologist who was doing a blind review of these samples, when he began looking at the liver tissue, his remark to me was, "Oh my gosh, this is textbook quality." And so you see a few of the slides that we reviewed together, normal liver, then in the middle, a slide of hepatocellular carcinoma, which is a liver cancer. And then a very rare type of tumor, angiosarcoma of the liver, is actually fairly abundant in the specimens stored here at SUBI. This review of pathology demonstrated 98% agreement between the diagnosis of the outside reviewer and the original diagnosis that was made at SUBI for these liver cancer specimens. 90% agreement was observed for the breast cancer specimens, and the real difference there was attributed to the lack of routine immunohistochemistry evaluations at SUBI historically. Next. In terms of the quality of stored blood, one of the ways that you can assess this is to look at the viability of the white blood cells, leukocytes, that you see in the first slide here. Another is the ability to thaw and grow stored lymphocytes. These were all demonstrated to be really excellent quality. In the third bullet, you see an assessment of cell surface proteins, which were measured very accurately. And in the fourth bullet, you see the ability to measure the serum proteins with great accuracy at least using ELISA for some key immunological factors like interleukins. The next. So now what I really want to spend most of the time now remaining is talking to you about a case study. This case study is meant to demonstrate that these biorepository materials are not only adequate, but they're actually outstanding to support modern molecular studies. And I'm going to tell you about a whole exome sequencing tumor DNA study from Mayak workers. Next. The Mayak worker cohort and its biorepository that we've been talking about is certainly a unique opportunity to collect some critical data about radiation mechanisms in liver cancer, as well as other cancers. But we took liver cancer because I have a longstanding interest professionally in liver cancer, so we might as well start with that one. The goal of the study I'm going to talk to you about now was to use whole exome sequencing to characterize somatic DNA mutations in these liver tumors that were stored at Subi. And I will mention now that the oldest of these specimens was put into storage 50 years ago. Yeah, 50 years ago. And the most recent one that we had in our hands was about 10 years ago now. Next. In the pilot study, we took 3 cases of hepatocellular carcinoma, 2 cholangiocarcinoma, that is cancer of the bile ducts, and 2 angiosarcoma of the liver, which I told you is typically an extremely rare malignancy, but there are about 10 cases of it in the biorepository. And we assessed DNA mutations in over 20,000 genes using modern sequencing technologies. Next. The DNA mutations that were found, this is not meant to show you great detail, but sort of a wide view of everything. The kinds of DNA alterations that we found upon sequencing, you see in the top part, is showing you that there are various kinds of deletions, insertions, and most commonly transitions. For example, the big red slice is where we have an A to G transition that has occurred in the tumor tissue relative to the adjacent non-tumor tissue. So that's the basic methodology here. And in the bottom, you see a comparison of different kinds of functional somatic mutations. So we detect, and this is very conservative calling of mutations, we found about 2,400 functionally significant somatic mutations. So these are not silent mutations. These are, when we say functional, they result in a change in the protein that is encoded by that gene. On average, there are 341 mutations per tumor, and the range goes up to about 766, as you see. Next. And what was interesting about these is several pathways. So we have, 2,000 genes, let's say, that are altered in these radiation-associated liver tumors compared to the adjacent non-tumor tissue. But recurrently, we saw chromatin remodeling alterations, the actin cytoskeleton organization, signaling pathways, and DNA repair pathways. The last one is what we expected to see because that's known from previous work on the molecular pathways that are induced by radiation, but we were surprised to see even some very more general things like the signaling pathways were altered. Next. And a side-by-side comparison of angiosarcoma of the liver on the left, cholangiocarcinoma in the middle, and hepatocellular carcinoma on the right shows somewhat shifting and different patterns of these altered pathways according to the type of histology. Next. Well, that's a way of saying that those preliminary data demonstrate the high quality of the tissue, that they're sufficient to generate these kinds of molecular data with state-of-the-art technologies. We have gone forward. That pilot study was published. We've gone forward. We have additional tumors. We finished sequencing another 15. And then just a few weeks ago, we did another 5. So there's going to be a grand total in the end of about 27 to 30 in our next upcoming publication, where we're going to try to confirm some of those early observations. Next. I do want to mention, as Dariya did, that there is a website where interested scientists can look and see. It's available in Russian and English. And they're able to go onto the website and review the metadata of the repository. That is a full catalog of specimens. The user can use the interface that we spent a long time programming, where you can shop for age, doses, your favorite types of organs that may be in there. You can see the number that are available. And these metadata do not have individual identifiers. That's why it's ethical to use it this way. And then users who want to obtain some specimens can go through and complete an online application. Next. And so I too would like to acknowledge those who came before, particularly the people that you see listed here as co-authors on the study. And special thanks, of course, to Tamara Azizova, who's the current director of the biorepository at SUBI, the founding director, Klara Maksinova, who I had the privilege to work with in my very first year that I went to Russia to meet with the team, and then also Evgenia Kirillova, whom I worked with for many, many years in great cooperation. You see a photograph of some of the same people that Dariya showed you. And I just want to say that I have seldom met such a dedicated group of people who really have done such outstanding work in maintaining a biorepository. So thank you very much for your attention.
Thank you so much for your presentation. And now I would like to invite another speaker, Ms. Lydia Zablotska, to present future opportunity severe pilot study. Thank you.
Good morning. Thank you to the organizers, ICRP and Dr. Rühm, for inviting me to present today. Yesterday we had a wonderful roster of presentations, and I really enjoyed the discussion that we had. These meetings are coming less and less because of the pandemic, but the Seversk study actually started from one of such meetings back in 2016, organized by Rampan in Würzburg, where I was seated next to Professor Akleev and Dr. Tahawa. And they told me about Seversk. Until then, I've never heard that name. And so this is how we started our collaboration. The first thing I did, I wanted to find out where Seversk was. And you might think, Chelyabinsk, Ozyorsk, but it's actually 200,000 kilometers deeper into Russia, Seversk from Chelyabinsk. It will take you 24 hours if there was a road, but usually people fly. I have personally never been there, but we were planning all these trips. So the story started in 2014 with many of you who are present here. I know Bruce and others went to Seversk with Barry Fontes and met the people from the Seversk Biophysical Research Center and looked at their materials, looked at the data quality, reliability, availability, completeness. They spent several days there, really, opening the books and looking at everything. They were particularly impressed that this cohort is different from Mayak, is because 98% of information and exposures are low dose, and completeness of follow-up at that time was 80%, and now it's 91%, as I'll show you. And so following that visit, a decision was the conclusion of the visiting committee was that JCCRER recommended that the study be initiated. For a number of reasons, that did not happen. And so when we met in 2016, I started collaborating with this group of investigators. And I was particularly interested in that because I work in Canada with uranium processing workers. And Seversk Biochemical Combine, as it was known at that time, has a number of plants, and one of them employs uranium processing workers, about 4,000. And so we invited the Seversk investigators to participate in our international pooled analysis of uranium processing workers. Unfortunately, we found out that they don't have a database completed or published, and no one knows about that study. And one of the inclusion criteria for the study was that you have all of that published. So I worked very closely with the investigators to publish the paper that came out in ICRP in 2021. Seversk is a closed study of about 100,000. Everything about it, organization, was geared towards this chemical plant, which is actually a combination of several plants. It was started operation in 1941 and became critical in 1950. Currently, it's referred, as of last year, it was SGCE. This is what you see on my slides because this is how it's referred in our documents. But now the name has been changed again, and it's called Seversk Chemical Plant, SCC. There are some of the pictures of the production of the plants. And the aims of the study that we proposed in 2021, where there was an open call for proposals, was to look at the doses, estimate the doses from uranium for participation in the IPOS study, conduct dose response analysis of lung cancer incidence in relation to external doses and estimated doses from uranium exposures. That was for a sub-cohort of 4,000. And Bruce Napier and Richard Bull were all ready to go. We signed the contract. But unfortunately, all collaboration, as you know, stopped in 2022. So I received the data for epidemiological research, but the urine analysis have not been transferred at the time. So the first two objectives were not possible to complete. They're sort of suspended. And I worked on the third one. So what do we have in terms of methodology? This analysis, epidemiological analysis that I'm doing, we have gamma radiation exposures from workplace badge measurements. There are, as I mentioned, individual internal exposures from uranium and plutonium could be estimated because there are measurements, both internal outpatient and inpatient in the hospital. Most of the workers that were exposed to these isotopes have about 1 or 2 measurements a year. So Bruce was telling me that this would be quite challenging, but possible to estimate these doses. And then in terms of outcomes, there is passive and active follow-up. I should mention in the discussion of methods that the group that in Seversk really benefited from the studies in the Southern Urals Research Center, because their plant became operational much later. So they learned from their experience. At the time I met them, all the data was computerized. The outcome, the follow-up was passive and active follow-up, used the methods that were used for Mayak and for Ozorsk and for Techa River. So there was definitely a carryover, very big effect, and they knew. So we didn't have to put as much work into forming this cohort. Also, very importantly, everyone who lives in that city of 100,000 people receives their care in one hospital. So it was easier to computerize the medical records if you have everything in one place. And even people who are retired still go to that hospital. And of course, in the 80s and 90s, a lot of people, when the city opened, left. But then many retirees came back to receive their really good care at this hospital. So that also helps with the follow-up. This is a picture from our paper that was published in 2021, where we describe how all of these databases came together. They're related by ID. And we have, in addition to occupational annual doses and information about their measurements in urine, there is also a lot of other information from their occupational record in the plant. There is also information from about medical and social factors from medical histories. For example, there's information on smoking, on education, on social status at different stages, how the people processed at the beginning, at the start of employment, and when they change job, they get additional education, something else. So all of that is kept. There's also a biorepository. That was the paper about that. It was published in 2015, if you're interested to find out. And it also, again, was very well organized and benefited from interaction with researchers at other Russian institutions. In terms of the follow-up, the databases are organized. We have a passive mortality follow-up, and there are also specialized morbidity registries. So for example, there is acute myocardial infarction morbidity. So anyone who has ever been diagnosed is entered into that database. And from then on, all of their laboratory values, if they're ever hospitalized or come to the outpatient clinic, they're all combined in that registry, which is very unique and would be very interesting to look at. There is also databases for other specialized diseases such as osteoporosis, thyroid gland diseases, and congenital malformations. This is the data have been given to us and shared with us in several updates. So the latest one that I have is the workers who were first employed from 1950 to 2010 and followed up until the end of 2018. The total number of workers is about 65,000, of which 25% are females. At the end of 2018, only a very small portion, about 5% of them were still employed because many of the plants have been closed. The average duration of employment is about 15, with a median of 11 years. The average follow-up is about 26 years. For some workers, we don't know what happened to them after they stop employment, but for many we do have, which is reflected in this long follow-up. The age of the start of employment was about 24, and the average age at the end of follow-up in 2018 was 59 years. The facilities that you see represented here are sort of big ones, and we worked with a number of these and analyzed them together and separately. But what's very important that we have a cohort of about 30,000 people from the so-called support facility, which have minimal or no radiation exposures and serve as an internal comparison. In terms of the birth cohorts, the majority of people were born from 1920 to 1960s, but there is also a very big proportion, about 20% of people who were born in the 1960s. So that shows you that you have 9 young workers that are still working probably and being followed up. We had some substantial issues with information on smoking because it came from the original intake when people started working. And that question was collected by medical professionals and was missing for a very big percentage. So, you know, when I did the analysis, I looked what was done by Dale and others, and we tried to look at the smoking information. But with that amount of missing, we went back and our colleagues rechecked it, but this is the best that we could get. To adjust for the socioeconomic status, we are using social status and education, which are much more complete, and we have multiple endpoints for this. This table shows the number of workers that were monitored at different facilities. So, for example, about 22% of workers from the enrichment facility were habitually monitored for external exposures. And about 12% of that have at least one measurement of internal exposures. And of course, the people at the support facility, only 10% of them were monitored. These probably were workers who were sent on repair work and given a dosimeter, so that's why the proportion is so small. In terms of the doses, the full cohort of 65,000 people, the mean cumulative dose is about 27 millisievert, but we have workers going up to 3.7, 3.6 gray. And then among exposed, which is much smaller, just a third of the cohort who have any kind of dose, the mean dose is about 82 millisievert. of monitoring for internal exposures. As I said, we have urine analysis, but not estimated doses. About 14% of the total cohort have at least one urine analysis. This graph represents the distribution of annual doses, and the bars show the number of workers for external exposures. And so you'll see that there were some sort of events at the beginning in the 1950s, but the real employment started where a lot of workers came to work was about 1960s. And the mean doses were not that high compared to other facilities in Russia. This table shows the distribution in bins of doses, and you will see that about 26% had any kind of dose, and it was less than 100 millisievert. And very small percentage of male workers had doses higher than 100 millisievert. And among female workers, that was just, a total of 2% who had a dose more than 100 millisievert. This very busy table shows, for those who are interested to know, the median cumulative doses, external doses, by among those who were monitored by facility. So, for example, if you look at the females, they worked at all facilities, enrichment, plutonium, and we have the mean doses for them. And of course, they were much lower. If you look at, for example, plutonium, their dose is 5 compared to 22 for males. And then we also see a very interesting sort of drop down in the median doses. If you look at the first year worked, goes in the 50s, it was 72. And then in the 90s to 2010, it was 4.2, the cumulative dose. And then also the age at start of work also, which of course represents people who worked more years and have a higher cumulative dose. By the end of follow-up in 2018, about 27% of workers have died, and the code of death is known for 91%. So we have 24.9% of people in the cohort who have died, and we have a code of death for them. Of those, about 6% is a cancer code of death. And then if you exclude liver cancers of the lung, liver, and bone to analyze it, because we don't have uranium and plutonium doses, that drops down to 27-30. We also have a very large number of CVD deaths. And then cancer incidence follow-up, which was done again through the hospital and regional and Oblast, Tomsk Oblast Cancer Registry, but also looking at the cancer registries in surrounding Oblast. We have about 6,200 cancer codes, and 5267 of them are excluding liver, lung, and bone. So as I mentioned, this is the latest submission of the data, but I analyzed the interim data and presented it at a conference 2 years ago. So that was a slightly smaller cohort of 61,000, and the follow-up for this one ended in 2013. We used the traditional methods of analysis and estimated these are risk for solid cancer incidence, excluding lung, liver, and bone cancers. This is an analysis showing categorical dose response. It is highly significant, both with linear trend and in terms of heterogeneity, and relative risk goes up. This is an estimate of excess relative risk that we see. So in a total, in a cohort, we see a 0.34 estimate of excess relative risk for all these solid cancers, excluding LLB. It's highly significant. There is the risk for males and female look differently, but they're not. But that difference is not statistically significant. Smoking status, as I mentioned, this is very highly suspect because of the missing data. And then there are some differences in estimates between different plants, but the confidence bounds overlap, and this difference is not significant. And lastly, the analysis of heterogeneity by attained categories of attained age and age at start of employment, and we don't see any significant differences there. So in summary of this interim analysis, this was the analysis of 4,000 incident solid cancers, excluding LLB. The dose response was significant. There were some differences between smokers, non-smokers. We don't really believe it because so much information is missing. But we can say that the risk so far did not differ by sex, plant, attained age of start of employment. And so this was the first analysis of incident data in this cohort. It is one of the largest and the oldest uranium processing complexes. There were only very few minor accidents and very few workers have very high doses. Extension of follow-up increased the number of cases in the cohort by 32%. And finally, just a few slides about IPO study that I mentioned is that also is being supported by the DOE. So we include a number of studies. The main criteria is that the data has to be published and data available for sharing. And workers have to participate in one of these 4 steps on uranium processing. And so we have now went through a very extensive harmonization process and have about 36,000 workers. We are very excited that we are able to harmonize exposures. Uranium processing workers pooled analysis has never been done before because the harmonization is so hard. And our dosimetry group with Estelle Devesne and dosimetrists from other individual cohorts have done a marvelous job in doing that. We have a very large number of cancers to analyze. And these are the names of my collaborators on IPO in France, Germany, UK, and USA. And Ashley is here, so you could ask her about that. And this is the funding agency. We also have great support from European organizations, in-kind support, and a very dynamic group of researchers. Thank you very much, and particular thank you for my collaborators from Seversk, who I understand is on the line and listening, to Dr. Tahoe, Milter, and Carpo. Thank you.
Thank you so much. Now we will have a question and answer session. Will be moderated by Benjamin French and Nolan Hatter.
I think we have about 20 minutes or so for questions, although that stands between us and the coffee break. And participants online are welcome to submit questions through the Q&A function. Yeah, back in the back corner, Gail. Yep, you do.
So I have a question for anybody that can answer this. When I was at SUBI, maybe about 10 years ago, certainly the focus here is on human samples, but SUBI also had samples from 30 to 40,000 animals, really unique. We actually worked with some of those samples, published a paper with some of the SUBI investigators. This was part of a Melody sponsored project. And I guess I'm wondering if anybody knows whatever happened to those tissues. Are they still there? Did anybody ever do any archiving of them? Because we have most of the old DOE archive, and boy, it would be wonderful to be able to compare some of those animals to some of our animals.
Anyone able to address that question?
Excuse me, can you hear me?
Yes.
As for the samples, the activity in order to sort of archive them and preserve them is sort of slow, but more or less moving ahead currently. And I may not say the samples are in say perfect condition, but they do exist, and there is at least some activity in order to archive them and make a proper catalog and proper preservation and all that.
Thank you, Mikhail.
Thank you for that response. Any other questions?
So this question's for Chris. Chris, here's my question. Of the samples that you've been able to do DNA work with, about what are they all in good enough order to get DNA sequencing on, or do you have to select in advance? I mean, we found with our animal archive that we actually have to run a few tests on our samples before we choose to sequence. So I'm wondering whether you're finding the same thing with your samples.
We are finding the same thing. And in our hands, about 1 in 5 fails those early tests, and then we don't proceed to the expensive sequencing. It really has to do with the amount of DNA, not so much the quality.
Thank you. We found about the same thing. And, you know, with samples that are 50, 60 years old, that's pretty remarkable, isn't it?
It's incredible. And, 10 years ago, people would say you couldn't do this. But modern techniques of DNA recovery are great. And then the new technologies to sequence are more sensitive than ever. And even those are improving.
Thanks, Chris. And thanks for staying awake.
Coffee will do that.
Yeah, a question from online to be read.
Yes, that's a question from Wim Itema. As shown during the wonderful presentations today and yesterday, inhalation of plutonium could lead to cancer or lung damage. Asbestos is known to have a globally similar effect, where either fibrosis or lung cancer can develop. In the past, the radiologists told me that they are mutually exclusive. Apparently, developing fibrosis after being exposed to asbestos means that lung cancer won't develop. Does inhalation of plutonium have that same effect? If so, would it be beneficial to see if any reason, other than personal luck, can be discovered to explain the different reactions? Anyone willing to answer?
That sounds like a Lydia question to me, but she says no.
This is Chris. I can say a few words because I've looked with the pathologist at these liver tumor tissues and the surrounding tissue. Only in a few cases did we ever observe fibrosis, and especially not cirrhosis. And those are typically going to affect the entire liver, as opposed to where the plutonium is deposited, some local effect in fibrosis. We didn't see it very much at all. So typically, those who have had hepatitis C for their lifetime or those who are heavy drinkers will have these very high levels of fibrosis in the tumors and cirrhosis. But in this set that we looked at from Mayak workers, we saw maybe 1 in 5 with those characteristics. And the rest seem to have very, very little histological evidence of any kind of fibrosis going on. So that's just a few observations. With regard to asbestosis, I don't know so much about that and how it would compare.
Thanks, Chris.
This is David Richardson, just to briefly follow up, and at least in the US, the opinion used to be the opposite. When they would do attribution of lung cancer among asbestos workers, there was an argument that asbestosis must be present in order to attribute the subsequent lung cancer to the asbestos exposure. But that's not true either. There's no necessary reason for those to be mutually exclusive or mutually necessary.
Thanks, David.
May I also add something to that question, to that answer?
Please. Yes, please.
Actually, plutonium-induced pneumosclerosis is completely different from, for example, asbestosis. Plutonium, the pathological basis of plutonium-induced pneumosclerosis is alpha particle damage to the blood capillaries, small blood vessels inside of the lung, and they're filling with connective tissue. As for mutual exclusiveness or mutual necessity for lung cancer and fibrosis, there is no evidence for that in Mayak workers cohort.
Thank you.
Thank you. I would like to refer to your presentation and in particularly to the report of microdosimetry in the lung plutonium. I wonder whether this is very interesting, frankly speaking, and I wonder whether we could extrapolate some of the findings to the case of radon, mutatis mutandi, of course. But it will be really extremely interesting because this has not been done by radon.
I'm not so sure I should address radon. It's possible because they're both alpha emitters. Plutonium tends to aggregate in the lung, as you saw in the autoradiographs, and accumulate and sequester, whereas radon is more the daughters of radon. Sure, yeah, the daughters of radon, yeah. So there could be. I have not personally looked. I've looked at radium and some of these, but not radon.
Okay. Thank you.
Someone else probably has more experience with that.
Yep.
Thank you. Alexander Ulanowski, ICRP Committee 2. Is continuing to the question from Abel Gonzalez. I would also note that it was nicely shown that the damage to the lung tissue is created by the particles in lungs, alpha emitters, which result in the highly inhomogeneous dose distribution within the organ. At the same time, epidemiologists work with the doses averaged for the whole lung as an organ. And that's my question, how was this this challenge of the particulate activity and highly non-uniform distribution in the highly heterogeneous organ was addressed, and whether it was addressed and how? Thank you.
Anyone willing to take that on?
Well, then probably you might want to allow me to try this again.
Please.
Yes, please, Mikhail.
Thank you very much. Yes, in the new dosimetry system, we do have dose estimates per different parts of lung. Alveolar region, big bronchi, small bronchi, etc. The problem
That's
is
what I was going to say.
the problem with addressing dose distribution between different parts of different structures of the lung is that in order to do this, we must know the origin of the tumor, let's say. And of course, it's clearly understandable that with lung tumors, it is often impossible to determine whether this tumor originates from AI region or bronchial or big bronchi or something like that, because at least at late stages, it's virtually impossible to decide. So that's why this is what makes it quite a difficult task, at least in terms of data which we have for Mayak worker cohort. I don't know if this information is available for other cohorts. Thank you.
Thank you. Yep, Sergey. Microphone.
It's like comment, more probably question to myself. Okay, when we're talking about non-uniform, highly non-uniform distribution of plutonium in the lung, so I'm a radiochemist, right? So what we did in UCR, we dissect, but I think Michael mentioned those big BB, small BBs, AI. So it was a part of actually bound fraction study. So and when it was a request from Birchall and groups, they said, okay, sir, do this way, and we're expecting to see basically, I mean, for me, distribution of the doses, distribution of the material, right? I'm once again, I'm measuring concentration. My thought on a distribution of plutonium was if the concentration is in different types of tissue is the same, that's a uniform distribution. If I will see different concentrations in different times, then non-uniform distribution. So anyway, when there was tasked to do those BBAI, so when we did it, and I can tell you, we did it 4 cases, and all of them, it's original one Alan told me should be 1, 10, 100. I mean, roughly. So they expected to go more in a deeper lung. And when we did analysis, radiochemistry results, and I publish it, it's 1, 1, 1. So, and when I publish the paper, I know the paper from a hand. I mean, see all those microradiographies, guys did it in Subi. And that's a question to me. So when you say non-uniform, yes, all those made on those little slides. And if you're looking on the slide, yeah, it looks like different. But when we go to, let's say, bigger region, let's say the whole BB, so it's maybe it's a macro distribution versus macro distribution versus micro distribution. But to me, it's at least what we are seeing in USTR once again. So it's not animals and people. So when we have it, our lungs is 30, 40 years post-exposure. But I would say I'm seeing uniform distribution. Sorry.
Excuse me, I probably should specify my question more clearly. My question was mostly related to micro distribution around the particle which emits a short-range radiation. It results in the extreme gradients of those within the tissue and then the extreme gradient of effect. And that's when we consider the outcome in the whole organ, which was created by the exposures in the very wide range of doses at the very small parts. So that's a challenge, and probably this is for future work. And this study really clearly shows that there are still scientific challenges to be addressed. And I see a colleague want to continue.
Gail's jumping out of her seat back there. Okay.
What we as the dosimetrists, what we transfer to the epidemiologists at this time is basically, we look at the 3 big BB, little BB, and alveolar, and then we weight them 1/3, 1/3, 1/3, and that becomes the lung dose. So we are still doing kind of the traditional definition of dose is energy per unit mass, and we use a big mass.
So I can't answer in people, but in dogs, we're actually doing some of those same things. And what we're doing is using X-ray microscopy to look at where is the plutonium versus everything else. And what we're finding out, for one thing, is that a lot of the plutonium is not in lung cells, it's actually in macrophage. Okay, so so whoever mentioned earlier about the immune response, that's probably very important. It's not just in lung, it goes to lymph nodes. And in fact, there's a huge amount in lymph nodes. And there is and the distribution depends on the form in which the animal got the plutonium. If it got particulate, the distribution is very different than if it's in soluble form.
Yep. Thanks, Gail. We have time for one more question. Yep.
I think this is a question to Scott. I was interested in the microdosimetry studies they did on Mayak workers. Did they look at whether the workers were smokers versus and level of smoking versus the extent of sequestration of particles in various parts of the lung?
Valentin Kokorikov had two categories for male smokers in Mayak. There were smokers and liars. And the reality is probably all the males they studied were probably smokers. And then the women were typically non-smokers, but they lived in an apartment with a smoker, so they were secondhand smokers. So, but I don't think they actually detailed that out. And that's a good question because Clara Suslova, we published a number of papers on the effect of smoking, and but we never looked. We should go look at those different lungs between the smokers and non-smokers relative to macrophages and lymphatics. Dogs have a different lymphatic system than humans, and the human is poorly studied, and this would be a good one for. Yeah, initially they used about, I think in their initial, their cohorts were not very big. Yeah, because of the technology was so labor intensive.
All right, we'll leave it there and enjoy our coffee break. Thanks very much.
As you already know, now we have an ICRP session. Now, the purpose of the workshop was to get an overview on all the scientific results which have been achieved and even ideas how one could continue in the future, to get an overview on those projects within this joint US-Russian effort, 30 years of efforts. And many of the results are scientifically fantastic per se, but it's more. It's also something, many of the results we need, we as ICRP now, to enhance radiological protection. And we would like to demonstrate this now in this session. We would like to show you how Committee 1 and Committee 2 are using data which come out from your projects. And this is now the purpose, just a reminder, this is the purpose of this session now. And I hand over now to Olga. She will moderate this session. Please, Olga.
Okay, thank you, Werner. We will now have 5 presentations, 10 minutes each, about the scientific achievements and their use in the ICRP publications. So we will start with Committee 1, and Dominique is already here. Please, Dominique.
Thank you, Olga. Thank you, Werner, for the introduction. Yes, I will try. It will be a very quick overview. Don't expect any discussion about the results, but just to show what are the objectives, what are the current works ongoing in Committee 1, and to highlight the importance of the results obtained from the social studies for these different task groups. So as you know, Committee 1 is dealing with the effects of radiation exposure on the cellular, biological, and epidemiological levels. The idea is to look at what is new from the literature and to propose some ways to include it in the best way as possible in the system of radiological protection. This slide presents the list of members, so I'm not going to get into this, but several of them are in the room today. And we have 2 members from SUBI, Tamara Zizova and Mikhail Sokolnikov, who are contributing to the work of Committee 1. The aim of Committee 1, as all the different ICRP committees, is to produce reports on specific items. And I would like to highlight especially publication 150 that has been published in 2021, which was dealing on cancer risk from exposure to plutonium and uranium. And Richard Wakeford was leading this group, and he will be presenting just after me. There are 10 different task groups ongoing in Committee 1 at the timing, which is a lot. And all of them have been launched and implemented with the aim to contribute to the assessment of radiation-related risks. Especially some of the task groups have been launched in the recent years to contribute to the revision and to the review and revision of what do we know for the elaboration of new recommendations in the next decade. I'm not going to present all of them. So some of them are dealing with cancer risk, some of them are dealing with non-cancer risk, some of them are dealing with the construction of the detriment, which is the indicator that is used by ICRP to build to reflect the level of risk associated to doses for stochastic effects. I'm going to focus only on 4 of these different task groups and to show the link with the results obtained from the socio-role studies. The first one is TG91. This task group is dealing with radiation risk inference at low dose and low dose rate exposure for radiological protection purposes. And I'm not going to go into details because there will be a presentation by Werner Rühm of the results and of the work of this task group. But clearly, the Mayak worker studies, it will be highlighted in the presentation of Werner, is of major importance in the reflection about the effect of dose rate and the consideration of dose rate in the system of radiological protection. The second one is TG118. This one is dealing with the relative biological effectiveness, quality factor, and radiation weighting factor. This one is added by Gail Boloshak, who is in the room. So see if you have questions, Gail is there. And also, clearly, these models, some of the results derived from the Mayak and Techa studies, especially from the elaboration of the dosimetric system that have been elaborated for the workers and for the general population, are of great interest in the assessment of RBEs in support to the radiation protection system. The third one is TG119, which is dealing with the effects of ionizing radiation and disease of the circulatory system and their consideration in the system of radiological protection. As you know, cardiovascular diseases have been integrated in the system of radiological protection as a deterministic factor in 2012 with a threshold at 0.5 gray. And there are many, many results, especially coming from the Mayak worker studies that are of great interest to analyze what is the current knowledge about what do we know about the relationship between the radiation exposure, especially in the low dose range, but not only, and the risk of cardiovascular disease. This task group is headed by Tamara Azizova. That's not by chance. That's because she has been working on the subjects for decades, and she's one of the best experts in the world to deal with this question. And the last example I'm going to take is about TG121, so the effects of ionizing radiation exposure in offspring and future generations. This task group is led by Prakash Ande and Richard Wakeford. Both of them are there today, so don't hesitate to ask questions. And again, this task group is dealing with the effects of in utero exposure and also of exposure before conception, what we call the hereditary effects. What do we know today, and is the current scheme for the integration of hereditary effects in the system fit for purpose or not? That's the question that are discussed in this task group. And again, the importance of the data that are available in the southern Ural cohorts. It has been presented yesterday. The super study and also population from Oslina are of great interest in the analysis of the risk for children of people that have been exposed to radiation exposure. So that's a very rapid overview. I think I'm still in time. But with this, I will give the floor for presentation of more specific topics. And we're going to begin with Richard Wakeford for population 150.
Construction. I encourage all of you to save your question to Q&A session that we will have after the ICRP presentations, as well as the audience that is listening to us online and watching online to send your questions to Q&A, and we will take them after the ICRP presentations. So, Richard, please.
Okay. Yeah, thank you. Well, as Dominique says, I'm going to talk about publication 150. You can see cancer risk from exposure to plutonium and uranium, which was published in 2021, and it's freely available at the ICRP or via the ICRP website. And here it is, and here's the authors from the task group. You will see some names there, led by Margot Tiermasch, and of course there's Mikhail down there, Dominique, Ethel Gilbert, John Harrison, and others. So we had to work for quite a while on this, and it was really the Mayak data that we were able to rely upon for the plutonium results. And I've just taken some quotes from publication 150 to demonstrate really the importance of Mayak to the report. Plutonium, two main studies, cohorts of workers employed at nuclear installations at Mayak in the Russian Federation, Sellafield in the UK. Sellafield is essentially the equivalent of Mayak in the UK. Analysis of Mayak worker cohort provides an estimate of the slope of the dose response curve for the risk of lung cancer, obviously very important. Lower levels of plutonium exposure, Sellafield worker cohort provides results that, within relatively large confidence intervals, are consistent with those for the Mayak worker cohort. Diplomatic wording there, because really it's the Mayak workers that made this, but it's nice to have Sellafield added in there. And results from the Mayak worker cohort also show an association between plutonium exposure and the risk of liver and bone cancers, but not the risk of leukemia, as we've heard, and again, important results coming out of Mayak there. Publication 150 details recent Mayak worker dosimetry systems, and these are obviously, again, we've heard about these, important, and we were able to rely upon at that time, the most recent dosimetry system, 2013, system developed for the joint analysis of Mayak and Sellafield plutonium workers. So we were able to do a pooled analysis of the Mayak and Sellafield plutonium workers under the EU SOLO project, and you can see here that we're relying upon the study of Michael Gillies and colleagues published in 2017. And then we were also able to develop a lifetime risk using the ICRP baseline rates for this, that is composite Euro-American male population, and derived again from the SOLO project analysis of Michael Gillies and the colleagues. So that again was a very useful contribution to the report from Mayak. So for the same INTEC, we were looking at cumulative doses, lung tissue, plutonium oxide, soluble plutonium nitrate and insoluble plutonium oxide, and again, acute and chronic exposure, but it didn't actually vary very much, as you can see there, 1.4, 1.7 per 10,000 persons. Depending on solubility and chronic or acute intake. And we made this comparison with the baseline risk for this composite population, Euro-American, of about 630 per 10,000. And then we were also able, quite usefully, to make this comparison with radon decay products, which was looked at in publication 115. And so we could make this comparison of lung cancer mortality per milligray. And you can see that for the radon right down the bottom there, it's not much different from what you get from plutonium, which is interesting in its own right. So again, we will also make this comparison with a risk estimate based on the lifespan study of the Japanese atomic bomb survivors, of course, mainly exposed to gamma radiation. So we could get this comparison of the relative biological effectiveness of alpha particles from plutonium deposition in terms of lung cancer mortality. And again, we can find this very useful result of that this RBE is approximately 16, and then make this comparison with radon, where you get a factor of approximately 14. So again, useful comparison here. And you will have recognized, I'm sure, that those RBEs are very close to the radiation weighting factor of 20 used for the purposes of radiological protection and alpha particles. So again, coming out of the Mayak data, very useful basis for establishing whether we're close to the right value for when taking this radiation weighting factor of 20 for alphas. And indeed, we do seem to be. Interestingly, there's this, of course, there is a drift factor of 2 tucked away in there in terms of coming from the risk estimate from the bomb survivors down to chronic exposure, low dose rated exposure. So if we were to not apply a DREF, then the RBE would drop to about 7 to 8 rather than the approximately 14 to 16 that we have with an implicit DREF of 2. So that's a quick blast through the publication, and it's the analysis, though, the conclusion, the analysis of mortality data from plutonium workers at Mayak and Sellafield, and that's, of course, mainly from the much more highly exposed Mayak workers, together with these absorbed dose estimates that we've been looking at in this seminar. It's permitted the derivation of the risk of lung cancer mortality in terms of the absorbed dose from plutonium, obviously, basically very, very valuable. A comparison with the previously derived risk lung cancer mortality in terms of absorbed dose from radon decay products. Again, a very useful comparison. Derivation of lifetime risk of lung cancer mortality per milligray. And an estimate of the RBE, the relative biological effectiveness of alpha particles in relation to lung cancer mortality, and comparison with this WR of 20, which is used by ICRP. So all in all, some pretty useful stuff that we'll be able to get basically from the Mayak data for the publication of this report. So thanks very much.
Thank you. That was Richard Wakeford with publication 150, which is available on our website for you. And the next presentation will be done by Werner Rühm, and it is the publication which is not available yet, but will be put for public consultation this year. And it is the result of the work of Task Group 91. Please.
Thank you. Dominique already mentioned Task Group 91, which is radiation risk inference at low dose and low dose rate exposure for RP purposes. And this is the current membership. And again, you see Tamara is here for good reasons. And many or some other members are here in the room or are connected online. Just to remind you that ICRP uses risk estimates from a high dose rate study currently, the atomic bomb survivors, and then to use it for the risk estimates for RP purposes, then there is what we call a dose and dose rate effectiveness factor of 2 is applied, already mentioned by Richard a couple of minutes ago. And TG91 is to review the current scientific evidence to focus on the low dose effectiveness factor, which is basically looking at the curvature of those response curves, and the dose rate effectiveness factor, but not on the DDREF. When we talk about low doses, we mean doses of less than 100 milligray, and low dose rates is less than 0.1 milligray per minute averaged over an hour. So we follow here the definition by Unskier. And I should also mention that this work, the task group is now working for more than 10 years, and I can tell you it's quite a challenge in 10 minutes to give you an overview on this task group and with a focus on contributions from Mayak and Techa River. So just a reminder, this dose rate effectiveness factor was introduced by ICRP in the 1980s, and at that time the data, basically animal models, they suggest values between 2 and 10, just demonstrating the uncertainties or the range of values, and it's animal data. And then Unskier says in 1988 that such a factor certainly varies very widely with individual tumor type, of course, and with dose rate range. But they use this also and suggest values, a dose range, values in the range between 2 and 10. And this prompted finally ICRP in 1991 to introduce the DDREF with a value of 2. But it was acknowledged at that time that to choose this value, a value of 2, is somewhat arbitrary, but it was felt to be conservative at that time. And Unskier in 2006, they didn't use this kind of approach, but they analyzed the curvature in the LSS data using an LQ linear quadratic model. And in this way, the LDEF was implicitly taken into account, but it is said in the report that this is roughly consistent with the RP approach by ICRP using a value of 2 for the DDREF. So this was very quickly a historical review. Now the work of TG91, when we started doing that, we decided to work a little bit different than ICRP worked previously. So we decided that we wanted to publish whatever comes out from our work immediately in the open scientific literature, rather than waiting for the final report to be published, which is not yet there. So we started with a historical overview, a review. You can find this in the literature. Then we review typical dose rates and doses in radiobiological and epidemiological studies. There are 2 papers, an older one from 2018 and a more recent one from 2022, which was part of a workshop of the Melody research platform in Europe. We reviewed molecular and cellular studies. This was basically done by Simon Bufler. We analyzed animal data, and this was quite exciting because when we started this, we noted that for the first time, 2 international databases became available, one in the US and one in Europe. So we were able to do pooled analyses of huge animal data sets, which was not done before, could not be done before. And we did that within the task group. And we published the results. You see three papers here, which came out of this effort. Then we also looked, of course, at epidemiological studies. And if we first go for the DREF, which meant that we tried to compare risk estimates coming out from the high dose rate study of atomic bomb survivors with epidemiological evidence from low dose rate studies. And we heard already yesterday that it was just in the recent 10, 20 years that those studies became available. And this included, of course, studies on the Techa River cohort and studies on the Mayak worker cohort. But this also includes other cohorts, such as nuclear workers, INWORKS, for example, or the 1 million person study in the US. So this was very exciting that we had now the opportunity to test whether the idea of such a DDREF value to be used in RP is really supported by epidemiological evidence. So we did a meta-analysis. Roy Shore, Linda Walsh were very instrumental. Tamara Sisu was there again on the paper. So we did a meta-analysis to compare matched risk estimates from low-dose rate studies with the LSS study, and matched means matched for sex distribution and age distribution. This was published in 2017. We also redid the approach of UNSCARE, which was done in 2006, and this was led by Mark Little. And Mark, he's here. He was also very instrumental in 2006 for UNSCARE to do that analysis. This is published, the results are published. We also had a brief look at biologically based mechanistic models because we thought maybe this is an opportunity to extrapolate from higher doses to lower doses using epidemiological studies, but using mechanistic models which are informed by biology. The review is there, so you can find that. And finally, we also looked just recently at the current epidemiological evidence on low dose rate effects again. So this was, in a nutshell, the work of 10 years of this task group. You can find it, and they are all listed on the website, on the Task Group 91 website at ICRP. So now let me come to the most recent work of the task group, and most recent means that this is even not yet published, but this will become part of the report of this task group, which hopefully will be finished this year. I will come to that at the end of my presentation. So I want to demonstrate why the Mayak worker cohorts and the Techa River cohorts are important for this work. And what you see here is we redid our meta-analysis, Roy, Linda, and others, and we included 29 human cohorts, 29 epidemiological studies in this work. And this included, of course, published work on the Mayak worker cohort and the Techa River population cohort. And if you look at the table, which is just copied and pasted from the draft report here, in terms of occupational radiation exposures and mortality, we include Mayak workers. And you see the papers, the published papers we used. And what we also did, I should mention that when we used the Mayak worker cohorts, it's immediately turned out that the risk estimates coming from them are significantly different than others. But there were concerns whether internal plutonium exposure might have influenced the risk estimates. So the task group approached Mikhail Sokolnikov from SUBI and asked him to redo their analysis, but just focusing on the workers from the reactor workers, and exclude the chemical workers and the workers in the reprocessing plant. And interestingly, the result was not significantly different from when taking the overall cohort. And so this helped us, this close collaboration with the ICRP task group, with our Russian colleagues to address this topic. Environmental radiation exposures included in this analysis includes, of course, the Techa River study I already mentioned, and we also include mortality and incidence data. But there are many more studies altogether, as I mentioned, 29 studies, including nuclear worker studies, Kerala study, environmental high radiation background level studies, and so on and so forth. And this is my last slide, I think. So what came out of it? And this is quite interesting. If we focus on the mortality studies first, and if you look at the first line, so we always compared matched risk estimates from those cohorts with the LSS data. And what you see, if we use all mortality studies, 24 mortality studies, then it turns out for the DREF, the dose rate effectiveness factors, we come up with a value of 2. This was surprising. But it also turned out when we used the non-plutonium Mayak subcohort, then it changed already. And then we did this exercise, leave one out approach. So we left out one study after the other and checked how this study influenced the overall result of the meta-analysis. And it turned out nothing happens if we leave out the 28 other studies. But if we left out the Mayak study, then the DREF goes down to around 1. And this already demonstrates how important this study is. Now, if we just focus on the incidence studies, 9 of those incidence studies, you see the values for the DREF between 1.4 or 1.7, but please also have a look at the uncertainties, the range of uncertainties, so they are still large. If we combine all the data, mortality plus non-redundant incidence studies, and again, you see if we have all Mayak data included, we end up with 1.9, something like that. And if we exclude the Mayak data completely, it goes down to 1.2, 1.3. And the last line shows you if we leave focus just on, time is over, give me 1 second. If we just leave out a focus on studies below 100 milligray, which means that we leave out Mayak and the Chernobyl data, then you see the numbers again, something between 0.9 and 1.3. And this was to demonstrate how important the Mayak data are for this kind of analysis. I should mention that this, I already mentioned this is not yet published, but we will have intense discussion in 2 days from now with Committee 1. And the critical reviewers of Committee 1 are Prakash Hande, who is here, and David Richardson, who is also here. And we are looking forward to discuss and hopefully after that finish the report. And with this, I would like to thank you for your attention. Thank you.
Okay, thanks, Werner. And that was Committee 1 on the effects with their 2 reports. Now we move to Committee 2 on dosimetry, and Alexander Ulanowski will bring us through the relevant studies that are used in Committee 2.
Thank you very much, Olga. Dear colleagues, I am very much honored to become today a voice of the Committee 2 and to deliver you the message and our common view to the Southern Ural radiation reconstruction and radioepidemiological studies. And as you know, probably that ICRP Committee 2 is devoted to the radiation dosimetry, and it implements indeed multifaceted activity in the field of radiological protection of people, the environment from natural and anthropogenic sources of ionizing radiation. Doing this, committee members, supported by the members of the associated task groups, contribute to development of dosimetric methodology for assessing doses from internal and external exposures to ionizing radiation, develop reference biokinetic and dosimetric models for human, animals, and plants, develop reference data, protection quantities, dose coefficients, contribute to development of ICRP recommendations, and of course advise other ICRP committees and task groups on the dosimetry-related topics. Few numbers just to tell you who we are. We are 18 people from 14 countries. We have 1 emeritus member and 3 representatives from the international organizations. In the total, currently, our members support activities in 9 task groups of ICRP, covering different aspects from related to dosimetry, to radiation risk assessment, to the medical exposures, to the application of the radiological protection practices. And some of these task groups have been already mentioned by the preceding colleagues who reported on their activities. Coming back to the main topic of our today meeting, to the studies performed in the Southern Urals, and trying to summarize how the Committee 2 as a dosimetrist, mostly dosimetrist, how they see these activities. First of all, we can say that these studies performed for the very specific sources covering the fusion decay products, including the operations with the nuclear fuel, accidental and operational releases, waste releases from radiochemical processing facility, other things, also the airborne releases of radiodyne and noble gases, related to sources of the longer-living radioisotopes like radiocesium, strontium, and actinides. With regard to exposure type, we see that these studies cover both occupational and public exposures. With regarding the pathways, we see it's an intake of radionuclides, internal intake, and also it's external exposures at workplaces, indoor and in the environment. Exposure periods, they span from the really short-term or even emergency cases to the long-term chronic exposure. So this means that these studies demonstrate us the wide and systematic application of the advanced dose assessment and dose reconstruction techniques. And these studies are known to use individual data, independent instrumental data for verification of their outcomes and findings. And that's why assuring the good quality of this confidence in this data. And also these studies is devoted to different cohorts covering workers, occupational exposed, and the population like Seversk residents or population in the Techa River and the living on the eastern Ural radiation trace places. So being short and just providing kind of a summary of our view, we can say that the studies in the southern Urals are extremely helpful for the C2 internal dosimetry aspects by contributing to the ICRP systemic biokinetic models for different radionuclides and especially including actinides and strontium-90. They provide information on radionuclide retention and distribution in the body, which is based on the, again, extensive studies done in the bioassay in vivo measurements, and as we heard already, provided by the tissue bank data. Important, what we already heard yesterday and today, that these studies are very advanced in dealing with all topics related to the uncertainty and variability of those estimates. They apply sophisticated techniques, and these all serve for the better validation and to verify ICRP reference human data. These studies are invaluable because they provide realistic scenarios, for example, for such fields like emergency dosimetry, related both aspects, occupational and population exposure. With regard to external dosimetry, the situation looks very similar, and we say that these studies provide us a very good and very helpful realistic scenarios for external dose exposures and different exposure conditions with relation to sources, relation to exposure geometry, time dependence, location factors, and many others. Again, as it was said for the internal dosimetry, here, sophisticated and advanced technique applied in the studies to validate methodology and validate the dose reconstruction values and develop sophisticated techniques to analyze uncertainty and variability of those estimates. This really invaluable, and it really contributes strongly to our activities and to our way of thinking and understanding of the things. And as I said, that also for emergency dosimetry, we can find very good examples and very good historical cases which can be helpful to further push forward the activities of ICRP in relation to emergency dosimetry aspects. So besides these pure technical and scientific aspects, we shouldn't forget about the important role these studies do in advancing the overall radiological knowledge and building the future expertise worldwide. And this is quite natural. It's well known to everyone that the long-term and highly professional international studies, like the studies in the Southern Ural, create new knowledge, they advance methods, they provide evidence and improve understanding. And all of these are to the benefit of the international community and particularly ICRP with its Committee 2, which I represent just today. we can also say that the experience which is gained during these studies, experience in dose assessment, dose reconstruction, can be also generalized by ICRP and presented in the form of a supporting guidance on application of the ICRP recommended data in real-life exposure scenarios. Here one can make just a very good example with the ICRP publication 66 on the human respiratory tract model, which was a very advanced, very detailed and significant publication. And this was followed up by the ICRP supporting guidance number 3, which provided the practical examples and helped people to properly use these scientifically based recommendations from ICRP. Another important thing to mention that the scientifically sound and long-standing research program provide opportunities for development of new scientists and specialists. And these would be the people who will support future activities of the ICRP. And these people will assure that the knowledge is not lost, it's preserved and transferred further and used in the future work. And this is an important aspect. As I said, it's not technical and purely scientific, but it's very important to consider as an effect of such kind of studies. And as a summary, I would say that from the view of the Committee 2, the outcomes of the dosimetric and the epidemiological studies in southern Urals can be regarded as unique, high-quality scientific data of global importance and applicability. These are valuable input to the activities of the C2 in the field of internal and external dosimetry of human exposed occupationally in emergency conditions and in the environmental exposures. Also, this can be regarded as a potential significant input to the activities of the C2 in the field of environmental dosimetry and doses for the non-human organisms. And with this, I would like to say thank you, finish my brief overview and message from the Committee 2, but say that stay tuned, because the very good and very practical and clear examples of what just was said will be in the next talk. And I don't like to interrupt with our moderator, but this is a kind of soft advertisement for the next talk. Thank you.
Okay, thanks, Alexander, for taking us through the Committee 2 work and how the data of Mayak studies is used there. And we will look for more deep into examples with Jokisch, who will tell us about the dose coefficients work.
So small sample size, but based on the speakers this morning, it appears Committee 2 is taller than Committee 1. Though I suspect the confidence intervals overlap, and perhaps somewhere in the middle is the reference individual, we hope. One of the things that I've observed both yesterday's session and today, and people have spoken to the importance of cooperation for this work between governments, between scientists, and cooperation from affected populations. So many of the studies that we've been discussing have benefited from individuals contributing their time for measurements and things like that. Like several others in the audience, I've had the privilege of spending 30 minutes sealed inside a steel cylinder at URCRM for a whole body count. And different probably for me, who understands the mechanics of what a whole body counter is, and is kind of excited about that opportunity to get in there. And probably not the same experience for someone who may have questions about, current health effects, future health effects, past health effects, and what might be inside their body. So I wanted to acknowledge that cooperation as well. So Alexander set me up very nicely, as have several of the talks from the investigators, You're going to see several of the studies and papers that have been talked about referenced here. I'm going to focus specifically on internal dose coefficients and limit my talk to the most recent ICRP dosimetry. And so these internal dose coefficients provide effective dose to a reference person per unit activity ingested or inhaled. And the current set of dosimetry we've organized into two suites of reports, right? The occupational intake of radionuclides, or the OIR series, which is now fully published, and doses to members of the public, or what we call the EIR, the environmental intakes of radionuclides. The first part of that is currently in press, and you can find the draft that went through public consultation is still available online. And EIR Part 2 just went out for public consultation about a week and a half ago, I believe. So the ones I've highlighted in blue here are reports where the science that's been discussed in the last 24 hours, you can find contributions, citations in these reports. And I'm going to hit a few specific examples. So I was kind of excited. I thought I don't get to give too many talks where I don't have an equation somewhere in the presentation. I thought, oh, I've got one. I've got a talk that doesn't have an equation. Then I was reviewing my slides this morning and I realized that the first bullet on this slide is an equation. It's just written out in words. So internal dose coefficients, fundamentally, they're the product of two terms. A source term, which describes where radioactive material is in the body as a function of time. And then a physics term that describes, okay, for a source in that location, how much dose do we get to a region? And really, the source term is determined by biokinetic models. We've heard about those, and the contributions from the Techa River and Mayak workers have really impacted ICRP biokinetic models. So that's the piece we'll be focusing on in this talk. And really for three elements: strontium, which we've heard a lot about, plutonium, which we've heard a lot about, and one that we haven't heard much about in the last 24 hours, which is americium. So this is strontium. We'll start with strontium. And a couple, actually 3 papers here by Shagana et al. That have really contributed to strontium biokinetics. So from OIR Part 2, I'm not going to read all these quotes, but I picked a couple of quotes out of publication 134. So I'm going to pick up in the second bullet. For example, the ICRP model predicts that 2.8 to 3.2% of whole body strontium-90 is eliminated each year at times 25 to 45 years after uptake to blood, compared with averages values of 2.7 to 3.2%, depending on age, in adult males of a Russian population exposed to high levels of strontium-90. So here's an example of confirmation that ICRP biokinetic models are conforming nicely with data. And of course, when you're sitting on an ICRP task group or a committee, you like it when multiple studies line up and agree, right? Doesn't always happen. And here we have an example where it does. And then the last bullet is also dear to my heart, because this is something that I think we need to improve on in the next set of ICRP dosimetry, which is sex-specific biokinetic modeling. So something else that we have from the Techa River studies is that they did observe sex dependency in strontium biokinetics. But in ICRP dosimetry, we assign male biokinetics to the reference female. So I think that that's an example of where we have good data in the future. The ICRP should use sex-specific biokinetic modeling. And then, yes, strontium's going to impact the strontium biokinetics from Techa River is impacting the doses to the public as well in the ICRP dosimetry system. So switching to plutonium, we've heard about, I think all three of these papers I saw cited yesterday. Describing plutonium biokinetics, and there are others. I couldn't list every paper here. Richard Bull gave a great talk describing the respiratory biokinetic model and plutonium and the bound state, and two parameters specifically that the that Mayak workers have helped inform the respiratory biokinetics for plutonium in the ICRP are that bound fraction and that slow dissolution rate. So I'll read this quote. In conclusion, estimated values of the slow dissolution rate from the human volunteer and long-term monkey and dog inhalation experiments are remarkably similar. Estimates from the USTUR and Mayak Production Association are considerably higher and lower, respectively. So here we have animal studies not agreeing with human studies, right? And this informing biokinetics for plutonium. And here's an example of data actually showing up in an ICRP report from these Mayak studies. Actually, two examples. So, and then finally, americium. So here are two papers describing americium-241 in Mayak workers, both of which have informed ICRP biokinetics. So this shows up in publication 141. Sokolova et al. Confirmed that applying the plutonium absorption parameters to americium resulted in overestimation of the americium lung burden by 48%. So, right, americium doesn't behave exactly the same as plutonium. On average, over 456 autopsied cases suggested slightly faster lung clearance for the americium than for plutonium. So, and with that, I'll just wrap up by saying that yes, several examples here of studies that have informed ICRP biokinetics that are being adopted and used right now. And thus, this is, I think, is a great example of international collaboration, international studies that are benefiting global populations when it comes to radiation protection. So with that, thank you very much.
Thank you. Perfect. I was a little bit concerned because there are 7 reports and you managed to go through them in 10 minutes. Thanks, Derek. Now we have 10 minutes for 2 complex questions or 3 easy questions. It's up to you to decide what you would like to ask. And we have microphone there. Maybe Sarah, I could ask you to help me with the microphone, please. So anyone? Yes, Abel Gonzalez, please.
Thank you. Well, in relation to the new study on dose and dose rates, I mean, this was very much welcome. I believe we have not considered that for many years. And we know that the dose rate in Hiroshima was very high, and we know all the problems. Now, it's a pity that we lose that opportunity of not addressing another issue that we have not considered. It's not only the dose rate, meaning the first derivative of those, but the second derivative of those, the change of the dose rate, which in Hiroshima was very high, positive and negative. And in the normal situations, including in Mayak studies, is basically 0. I know that it's difficult to address this issue, but at least we have put clear that this is an open issue for the future.
Would Werner like to answer that? I guess the people listening online can't see you.
At least I don't know many studies on that so far, but if you have some, please let me know.
Okay, thank you. Sergey, please, next question.
That's a question to Derek. So Derek, those americium biokinetic studies, so it was based on pure americium intake, or americium was a part of plutonium decay?
Oh.
Okay.
Thank you. Just on the microphone. Okay, for the people listening, and the answer was both.
Thank you. Any other question in the room? No, we have one online question. Sorry. Appreciate the tremendous work done during the last 30 years on effective research in developing a sustainable platform to assure better lifestyle facing with the radiation. Now, how could the regulatory bodies and NGOs, namely radiation protection societies, assist in data collection and contribution to the related research? Could uniform methods be developed in this regard? So there's a lot of experience on collaboration. Would anyone like to answer that question? How could NGOs and regulatory bodies contribute to further collection of data and research? Bruce, would you like to say something? You look like.
I don't know too much about how the NGOs can do it. The nice thing about the regulatory bodies, especially through an activity like the JCCRER, is that they brought together many different national regulator agencies. I mean, we had EPA, we had NRC, we had NASA, we had National Cancer Institute, all contributing money and staff to doing this. NGOs typically don't have that much money, so.
Yes, and Werner would like to add a little bit to it.
Yes, I would like to speak for one of the NGOs here in the room, which is ICRP, which is an NGO and a charity, so we don't have money for that. So how can we support that? I think what we can do is we can show how useful those data are, scientific data, and how we can use them for the benefit of all. And this hopefully motivates funding agencies to put more money into the system, because in the end, really, it's all is upon money. Yeah? Most of it.
And willingness to cooperate, I guess. Yes, Dale, please.
I think ICRP is doing some work that sort of contributes toward this goal of helping people do this stuff. And in the world of detriment calculation, because it's an ICRP concept, I think for lifetime risk calculations, there's a real need for tools that people can use that sort of standardize. And I've spent a lot of work by one of the Committee 1 task groups on that. And supporting that by doing this work is very important. And then maybe that would encourage organizations with money to support that kind of activity. And the same goes for analytical methods and helping standardize, make those available.
Thank you. Yes, Bill?
Yes, I was very encouraged of hearing these talks, and because we're dealing with an extremely complex set of models when we talk about plutonium dosimetry. Now, one of the things that I still get confused on is when we talk about this bound state of plutonium in the lung, which, as I understand it from these talks, that really means what are regarded as sequestered particles in different regions of the lung. Now, when we at least make an attempt to, as I understood them, the discussions here, to calculate separate dose terms for those different regions. And I wondered how much biological evidence do we have for how long the particles and the dose accumulates in those, I think, 3 separate regions now. Is there actual biological evidence that differentiates them?
Who would like to take the responsibility for answering? Yeah, okay, good. Richard will give us an answer.
I'm not sure if I'll give you an answer, but I think basically the evidence for presence of a bound state is really driven by the Mayak autopsy data. I'm not familiar enough with the details to say whether they've looked at they must have looked at the different lung regions and concluded that, that there's evidence for a bound state in all 3 regions, but I'm not that familiar with the details of the data.
Looks like Sergey has something to add to that.
I'm kind of going against myself because UCR data were used to define this bound fraction, but what we recently published with people from Los Alamos, with Depeche Online, yeah, so we published 4 papers, and we had 4 cases from UCR analyzed. It was 2 cases when we could not, no matter what we are doing with bound fraction, we could not fit the data. So, but then Depeche, so he proposed scar tissue compartment, and with the scar tissue compartment, everything was fine. So maybe it's not a bound fractions, maybe it's a scar tissue there. So, but at that point, bound fraction usually, if you don't account for that, you underestimate your dose, right? So if it's a scar tissue, it looks like it's opposite way around. You have a scar tissue which is surrounding the particle, does not expose to your tissues, and you have a concentration activity there, but the effect from those particles is less because you have a scar tissue around. So, and yeah, and
Don't feel pressure.
Yes. So, and for the regions, yeah. So those, because as I said, concentration is 111 is the same, but the weight of those regions is different. So the dose distribution of the dose it's higher in parenchyma in AIs because the weight of that compartment is higher. I mean larger. So if you do distribution, basically there is a distribution of material versus distribution of dose in the lung. So material, to me, material distributes evenly, but dose is not evenly distributed in the lung. Something like that. It's my dummy thing.
Yeah, thank you, Sergey. I think we will close the Q&A session now, and you're very welcome to continue discussions afterwards. We will have now one bonus presentation by Ashley Golden, who will tell us about archiving the data. 10 minutes, Ashley, please.
I like that bonus presentation. This was something that the Department of Energy asked me to give you all a little bit of information. It's still in progress. It's still a lot of things in here are hopeful, I'll say, and depend, of course, on what sort of agreements can be reached appropriately for the process of archiving and making available potentially the data. So I'll go through this really quickly. The U.S. Department of Energy has a program called the Comprehensive Epidemiologic Data Resource, or CEDR. Roy Shore actually mentioned this yesterday in his presentation. And this has been in place since the early 90s and was in response to recommendations to increase the transparency of the human health studies that were being performed by or funded by the U.S. Department of Energy when it comes to radiation health effects. It is a data resource, and I think on the next slide, yeah, I have a QR code there, so you can take a photo and go check out the website. But it is free, free data repository with de-identified data. I'm going to tell you more about that in another slide. Over 1.5 million workers, more than 400 data sets. There's a unique bibliography that has more than 4,000 citations in it related to the work that the US Department of Energy has done since the 70s related to health studies, and a lot of other historical documents that can be difficult to find in other areas. Not a lot of users, maybe about 100 per year, that actually sign up to obtain an account, which is necessary to download any data. But you can go and just look at the website. You can, for any data set, look at the first 10 rows so you get an idea of what's there, which is really useful. So when we talk about how we protect this data now, there is an account requirement. All accounts are reviewed and approved by the Department of Energy, and we re-verify those accounts annually through a password protection system. And we are currently working on a more enhanced data use agreement. There's one there currently, but it's very simple, very simplified. And we are also working on a tiered access approach, which I'm going to tell you more about. As I said, data are de-identified. They use a pseudo ID that is created by my team. We mask all of our dates by using the 15th of the given month and the given year associated with that date. And we also will suppress data variables based on the request of the submitting researcher, of the submitting principal investigator, or if for outcomes we have less than 5 or 10 in a certain category. Okay, so why am I here and what are we being asked to do? So the first goal is to make sure we archive all this incredible work we've heard about over the past day and a half. Already, we have 380 manuscripts that we've received from PIs and from the program manager, Dr. Zoe, and we are making these available through CEDR for download. We do have some cleanup to do on these, but we should have that done here soon. There is a section under the website which might not actually be active at the moment because we were waiting to try to finish all the PDFs. But it will be under bibliographies, DOE Russian health studies. As well, we created a library for the Department of Energy that is not public access, for which all of those already exist. What we'd like to do, the hopeful part of this, is where appropriate to archive this data for Department of Energy in a non-public repository, and then where appropriate, where allowed, make some of this available to the public using the current framework of CEDR. And there's a lot of considerations needed there, a lot of approvals that are needed, of course, but we would just like to start thinking about it and talking about it and understanding what would be the most appropriate way and approaches, such as masking, categorization, aggregation, per person timetables, and so forth. This is a little bit of detail about how we intend to work with at least the US principal investigators. As of now, we've already started these communications and discussions. And this is a lot of detail on this tiered access approach that we're implementing. And I want to make a point, we still want to keep ease of access to this repository because there is a strong desire for the data to be used for educational purposes and classroom instruction. So many data sets are nice and square coming out of textbooks, and they don't really teach you the things like data cleaning and manipulation and other things you have to do when you're doing your own research studies. So we have found that this data is really useful for those kind of activities. We had a junior high school teacher who was using this data for unit conversion, and they were using the radium dial painters from the United States who painted the watch faces with radium and licked the brushes. And we introduced a whole new generation of students to the potential health effects from radioactive materials. And they were very interested and asked us to come and speak to them about that study and about that cohort. So level 1 will be for educational purposes. It'll be simplified data use agreements, a simplified application process. It will only include data sets where all individuals are deceased, for which human subjects protection regulations don't apply, or aggregate data or simulated data, which was an idea that Dale Preston gave us of a possibility that could go here. This data doesn't include demographic variables. We will leave maybe year of birth so that age can be calculated, but we would be excluding all other demographic information. When we move to tier 2, this is for more low to medium risk data sets that are going to be, the intention is to use them for research purpose. This will require an institutional review board determination, either by the DOE institutional review board or by the researcher's institution. It will be limited to the data that are requested, data and variables specifically that are requested. And it will have all the demographic data if that is part of the request and the request is approved. Tier 3 is probably the thing of most interest, I think, here, because this will be both the institutional review board requirements as well as approval by a designated owner of the data set. And of course, this is in addition to DOE's approval. So this would allow principal investigators and researchers to have some control over how that data is being accessed and utilized. So why does this matter? I kind of mentioned it before. Really incredible, valuable research results and data that we'd like to first preserve for potential future uses with the archiving process, and then possibly make available to others who would like to continue this work and to formulate new questions, new research based on the data. Kind of separately for the United States, the federal government, there is a policy to ensure access to federally funded research. So for Robin's sake, I put that there, that this helps them comply with that policy by making data from their programs available where appropriate and so forth. So that was fast. And again, it was just to generate some discussion and get people thinking about what would it take to make this happen if we are able to do more than just archive. So thank you.
Thank you, Ashley. And that brings us to the end of our day. And before I give floor back to Werner, I would like to say that there will be lunch served today again. And since there are several groups being served lunch, so we are encouraged to quickly proceed after the closing of the session to canteen or to the restaurant. And I give the floor back to Werner.
Well, you should go quickly to lunch, but first you should listen to my personal reflections of this workshop. So first of all, thank you very much for contributing to this workshop, which was quite a challenge, I can tell you, to organize it during these days. What I took from all the presentations, above the numerous scientific results presented, is the overall deep respect of all the involved scientists, which they shared with each other, independent of their nationality. We heard scientists presenting their results, and as you know, it's in our DNA as scientists that we raise a question and then we try to answer the question to solve the problem. But then, of course, as it happens, there are 5 new questions we raise, at least 5. So this is why science moves forward, and this is why we need to have more scientific efforts, also in the context of this workshop, of these studies. There's a lot to do to continue this research. This is another lesson I learned from the day yesterday and from today. The results presented at this workshop demonstrate how important international collaboration is for science, and specifically, of course, it also demonstrates how important it is for ICRP, because the ICRP strives to improve radiological protection for everyone exposed to ionizing radiation, independent in which country or region of the world he or she is living. Unfortunately, currently, the importance of international collaboration appears to be increasingly underestimated by some. The results we heard yesterday and today at this workshop clearly demonstrate, at least to me, that all these great results could have never been achieved by just one institution or just by one country. They were only made possible because there was an overall international collaboration across borders, political systems, societies, etc. it seems to me that it is our responsibility as scientists to reiterate and to emphasize this again and again. That is how important international cooperation is for scientific progress. And it's definitely important for the work of ICRP. So what's next now? Currently, as far as I heard, the scientists who came together at this workshop and the organizers of this workshop are exploring how to document this workshop and how to publish still unpublished scientific results. So if there is anything we as a poor NGO can do and help, please let us know and we will support you. This workshop was, at least in my perception, my personal perception, another milestone in the 30 years history of Southern Urals health studies that have been performed jointly by institutions from the US and from the Russian Federation. And please allow me to reiterate what I had already said in my introduction yesterday, that there were other projects funded by additional parties, such as, for example, the European Commission. I sincerely hope that this is a milestone, this workshop is a milestone, and that this is how this workshop will be considered or perceived in 20 years from now, after 50 years of continued, hopefully, continued studies. And we heard a lot of ideas what could be done in the upcoming years. At least this is my hope for the future. So I hope to see you all again in 20 years from now, at the latest, and discuss new results coming out of those studies. So with this, I would like to close this workshop. On behalf of ICRP, thank you very much for participating, either in person here in Vienna, in Austria, or online, wherever you are. Thank you very much, and have a safe travel back. Thank you.